Okay, ladies and gentlemen, you are all welcome. So in this video, we are going to look at the embryology of nervous system. So don't forget to subscribe, follow, and join my YouTube community for more videos. Introduction to nervous system embryology. So embryology of the nervous system, it involves a complex processes starting from ectoderm layer during early development. So generally, ladies and gentlemen, after fertilization, a zygote is formed, and of course, from the zygote, we have morula. As you can see, there is a full stage. After the full stage of cleavage, then of course the eighth stage of cleavage will give us a total of 16 to 32 cells, where we have a morula. And then of course, from the morula, we have blastocyst, and then of course we'll have embryo. So during this journey of life, of course, we can harvest stem cells at the zygote level. You can harvest stem cells from morula level, and of course even at the blastocyst level. So generally at the blastocyst, these stem cells are divided into three. We have mesoderm, we have ectoderm, and then we have endoderm. So the nervous system is a system that originates from the ectoderm layer. So as you can see during the early development, the ectoderm layer, as you can see from here, it is a layer that are used to drive the nervous system. It's a complex process that involves the formation of a nervous system starting from the ectoderm layer during early development. So it is essential for understanding congenital anomalies related to the nervous system. So what I want you to understand at this point, ladies and gentlemen, in this video, we are going to look at how nervous system are generated even at the stage of the embryo, because we have stem cells that have the ability to differentiate into each and every cells in the body, each and every tissue and organ in our body. So of course, ectoderm layer, it is one of the division of the layer of the stem cells that are used to generate nervous system. So that is what I want you to understand at this point. Nervous system are generated from ectoderm layer stem cells. So, and also very importantly, the development proceeds through multiple stages, include induction, proliferation, migration, and differentiation of neural cells. So if you ask how this ectoderm layer differentiate into the nervous system, it involves a multiple stages. And you shouldn't forget about the stages that are actually involved in the generation of the nervous system from ectoderm layer. This process includes number one, we have induction. The ectoderm layer has to be induced for them to initiate the generation or the formation of the nervous system. And of course, after induction, there is proliferation, we have migration, and then we have differentiation of the neural cells. So we have four stages, please. Don't forget that. The stages in which ectoderm is converted to or is used to the nervous system. It includes induction, it includes proliferation, migration, and then lastly, differentiation into neural cells. So then now, let's look at the development of neural plates, how neural plates are generated. So neural plate forms from ectoderm layer during the third week of embryonic development. So during the third week of embryonic development, the neural plate are formed, of course, as we can see from the ectoderm layer. So neural induction is triggered by signals from the underlying notochord and mesoderm. So generally, the formation of this neural plate is induced by the signals, and these signals are coming from the underlying notochord and mesoderm. And of course, these neural plates are elongated and begins to fold, leading to the formation of the neural tube. So you see, first neural plate is first generated, as you can see from here. This is where we have the neural plate. So neural plate will first be generated. And how these neural plate are generated is through signaling. And this signaling is coming from underlying notochord and mesoderm. So after this neural plate is generated, and then the next thing, the neural plate will elongate and then begins to fold. You see, this is where the neural tubes begin to fold. So next thing after the folding, then it will lead to the formation of neural tube. And as you can see from here, the neural tubes are generated by folding of the neural plates. So I hope you understand at this point. So first, the neural plate is developed. So after the neural plate is developed, and then it will elongate itself, and then later on it will fold, which lead to the formation of neural tube. So then the next thing is then the formation of neural tube, how this neural tube is formed. So as you can see, this is exactly what I explained. So you see, we have neural plate. After the neural plate is formed, and then of course, there will be elevation or elongation. After this elongation, as we can see, the neural plate folds to create the neural groove. So neural groove is first generated, which then closes dorsally to form the neural tube. So see from the neural tube, from the neural plates, it will generate neural groove. And then the next thing, it will dorsally closes itself to produce neural tube. So the closure of the neural tube begins at the future cervical region. So at the future cervical region, that is where the closure of the neural tubes begin, and then proceed both cranially and then caudally. And of course, you know that there are certain diseases that actually occur or that happen as a result of a failure of the neural tube closure, resulting in a congenital anomalies such as spinal bifida and anencephaly. So you should understand after the neural plate is formed, then there will be elongation and then elevation, then there will be folding, and after the folding, then there will be closure. And then this closure of the neural tubes is begin at a future cervical region, and then it will proceed cranially and then caudally. So this is what you need to understand. So then the next thing after the formation of the neural tube is then the differentiation of neural structures. So the neural tube, after the neural tube is formed, so the neural tubes will differentiate into central nervous system. And this formation of the central nervous system, it includes the formation of brain and the spinal cord. So it means that the brain and the spinal cord are generated or are formed from the neural tube. And then the cranial part of the neural tube would enlarges and subdivide into primary brain vesicles. So as you can see from here, ladies and gentlemen, we have three primary brains vesicles generated, and these primary brain vesicles that are generated are called, which include forebrain, midbrain, and hindbrain, which is called prosencephalon, mesencephalon, and rhombencephalon. So these are the three primary brain vesicles that are generated from the cranial part of the neural tube after the enlarge. And then, of course, as you can see, the forebrain will further be divided into two, telencephalon and diencephalon. And of course, for the midbrain, it remains as it is, which is mesencephalon. And also at the same time, the hindbrain is also divided. It's further differentiate into two, which include metencephalon, and melencephalon. So this is how they will keep dividing until they fully differentiate into the brain structures. So as we can see, even the telencephalon, it will further divide. As you can see, it started from three primary brain vesicles and then finally the five secondary brain vesicles. And at the end, you can see that the telencephalon will generate cerebral. And then the dicephalon will produce eye cap, thalamus, hypothalamus, and epithalamus. And of course, the midbrain remain as the midbrain. And then for the metencephalon is further differentiated into pons and cerebellum. And then the melencephalon is differentiated into medulla oblongata. So this is how I want you to understand that the neural tubes is further differentiate itself into the central nervous system, which include brain and the spinal cord. And of course, the cranial part of the neural tubes is the one that enlarge and then further subdivide itself into the primary brain vesicles, which started from three and then finally to five, where the different part of the brains are generated. And then, of course, as you can see, also the caudal parts of the neural tubes develops into spinal cords with specific regional differentiation. So these are actually the two different parts of the neural tube, which differentiate into the cranial part differentiate into brain vesicles, while the caudal part of the neural tubes differentiate into spinal cord with specific regional differentiation. So then the next thing is actually the development of brain vesicles, which I explained earlier. The primary brain vesicles forming during or formed during the fourth week of embryogenesis, and it includes the proencephalon, mesencephalon, and rhombencephalon. And these vesicles will further subdivide into secondary vesicles and giving rise to various brain structures as we discussed: cerebrum, eye cap, thalamus, hypothalamus, and epithalamus. So you see, you see, this is where we have, it started from three primary vesicles and then to five secondary vesicles, and then from the five secondary vesicles, then it will further differentiate into the following brain organs. So, and then the brain continues to grow and develop specialized regions responsible for higher functions. So then the next thing after this is then the formation of neural crest cells. Neural crest cells originate at the border of the neural plates and epidermis during neural tube formation. So as you can see, the neural crest cells are originated at the border of the neural plates and epidermis during the neural tube formation. And generally, these neural crest cells, they migrate extensively to form a diverse structures, including peripheral nerves, ganglia, and then melanocytes. So as you can see, the peripheral nerves, the ganglia, and melanocytes are originated from the neural crest cells, which actually originate from the border of the neural plates. And then, of course, this neural crest development is crucial for proper formation of the peripheral nervous system. So you can see that peripheral nervous systems are generated from neural crest cells. And then, so the next thing is then the differentiation of the spinal cord and the brain. We have what you call neural progenitor cells, which differentiate into neurons and glial cells through a series of precisely timed processes. And then the ventricular zone in the neural tube. Of course, in the neural tube, we have ventricular zone. So this ventricular zone in the neural tube is primary site for neurogenesis during early development. So this is where neurogenesis takes place in the ventricular zone. And then the axonal growth and synaptogenesis occur at the nervous system matures, as the nervous system matures, establishing functional neural circuits. So you see the axonal growth and the synapses, that is the formation of the axon and the synaptic clefts or synaptogenesis, it occurs at the as the nervous system matures, establishing functional neural circuits. So then the next thing is, of course, congenital nervous system anomalies and summary. So disruption in embryonic development can lead to anomalies such as spinal bifida, anencephaly, and holoprosencephaly. So understanding embryogenesis helps in diagnosing and potentially preventing some congenital defect. The embryology of the nervous system is a complex but well-coordinated process essential for normal neurological function. So, ladies and gentlemen, this is where we are going to stop. Don't forget to subscribe, follow, and join to my YouTube community for more videos. See you in the next video.